Action. Here we are in a cotton field. I know it's really a classroom, but I want you to use your imagination. Let me introduce you to a little fella named Palmer. His full name is Palmer Amaranth. Some folks just call him a pigweed because he's not supposed to be in this cotton field. He doesn't care. He's just going to live his life the best way he knows how.
I'm a little seedling, short and stout. Here is my leaf, though it's barely just a sprout. When I get all grown up, I will be the biggest weed you've ever seen.
He's just a little tyke right now, but give him a few days of warm sunshine and he'll be the biggest terror of this cotton field.
Hello, cotton plants. My name is Palmer Amaranth, to be exact. Here's my brother Palmer and my other brother Palmer. Our parents named us all Palmer. We're all closely related, and pretty soon, if something is done to stop us, a few thousand of my other brothers will end up. We'll be up and out of the ground, ready to dominate this ecosystem. The cellular process making us grow is called mitosis. We love mitosis. Make my cells go through mitosis. Make them develop again. Our chromosomes get big and then replicate. I don't care if they super mutate. Oh, we'll root for our DNA. If they don't win, it's a shame. Because it's 1, 2, 3 splits, we're done with mitosis.
It is.
That may be what the plants are thinking, but let's see what's going on inside of the cells. We have the cell wall here, cell wall. Within the cell wall, we have the nucleus. Hey, cell, what have you been up to during your interphase?
As leader of this cell, I've been getting ready for the big mitotic event. This cell has been growing and doing all the normal functions of living. My cytoplasm has been expanding. I have a lot more organelles now. I even replicated my DNA. This cell of mine is totally stoked and ready for mitosis.
I see that you're ready indeed. Within the nucleus, chromosomes are starting to condense and coil. Chromosomes coil. They're coiling. This stage is called prophase. Well, we're just going to watch a single chromosome. Remember that within this particular species, there's 15 other chromosomes doing the exact same thing at the exact same time. The chromosome that's already replicated, so it has chromatids attached to the centromere. The nucleus membrane is starting to break down now, which means it's lowering, and the spindle apparatus begins to take shape, the imaginary spindle apparatus. We now enter metaphase. The nuclear membrane is now completely gone and out of the cell. The membrane can get out of the cell. There's spindle fibers attaching to the centromere. The chromosome orients to the equatorial plane, so they're about halfway through in the middle of the cell. The chromosome is at the most condensed stage at this point. Now it's the best time to karyotype the chromosome. So smile for the camera. Those are some pretty ugly smiles. Okay, now we're ready for anaphase. This will be the quickest phase of mitosis. The centromeres separate. Each chromatid is by itself and called a chromosome, a daughter chromosome at this point. The spindle fibers are pulling the centromeres towards the end of the cell, and the homologous chromosomes will go to the same location to the opposite sides of the cell like you see here. Finally, telophase has arrived. The daughter chromosomes have reached the opposite ends of the cell, and they start to uncoil, and the nuclear membrane reforms. However, this time there's 2 nuclear membranes forming around each set of daughter chromosome cells. So we need another set of nuclear membrane. Nuclear membrane leader, assemble the next nuclear membrane. We need the other 2 participants who are nuclear membranes to surround the chromosome. The cell wall, we need the other participants who are the cell walls to reform an additional cell wall. Where are my other cell wall people? final division is called cytokinesis. Now we have two identical cells that are ready for life in the interphase.
Wow, mitosis really worked wonders. I am now 3 feet tall. I have survived wind, rain, drought, disease, birds, herbicides, and cultivators. I am now ready for some serious action. Hubba, hubba, hubba. That has to be the prettiest plant I've ever seen. She's only 3 rows over. Actually, she's the only non-cotton plant I see. The farmer has done a deadly job in killing out most everything but his cotton. Hey, you weedalicious plant. What is your genus and species?
I'm surprised you didn't recognize me. I'm the same species as you, and I'm a girl pigweed. Didn't you know that we are dioecious species? I only produce female gametes, and you only produce male gametes. Ha 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 ha. Ha.
While each of these plants specializes in zygote formation, the process of producing these zygotes is fairly similar. Now let's go back to the cells for more details. Hey nucleus, we're back again, and we'd like to see the process of meiosis. Please tell us the importance of meiosis.
Without meiosis, each generation would have twice the number of chromosomes as the parents. It allows the maternal and paternal chromosomes to randomly assort into each gamete. During meiosis 1, crossover occurs that allows for unique combinations of alleles in a new chromosome.
Like mitosis, the first stage is called prophase 1, only it's a bit more complex. There are actually 5 stages within prophase 1. The first stage is called leptoteme. The chromosome appears as a long thread. And during zygoteme, the homologous chromosomes pair up and there's lateral association called synapsis. Crossing over recombination occurs in the next stage called pachyteme. This crossing over event provides increased genetic variation. Understanding this concept will help us understand segregation ratios and traits in the inheritance of principles first proposed by Gregor Mendel. So let's have the chromatids actually cross over. As you can see now, there's chiasmatas in there, the places where the chromosomes are in contact and can exchange genetic material at that point. We now enter diploteme. The homologs begin to separate, and the last series to separate are called the chiasmata, which provides evidence of recombination. This last stage is called diakinesis. At this point, the chromosomes shorten and condense, and the nuclear membrane disappears. Nuclear membrane disappeared. We are now at a metaphase in which the chromosome is aligned at the equatorial plane. The chromosomes become attached to the spindle apparatus. Next up is anaphase 1. Unlike mitosis, the sister chromatids stay together or remain joined to the centromere as they're pulled away from their homologous chromosome. So this chromosome all needs to go to one side as they're being pulled towards one end of the cell. We've now reached telophase 1. The chromosomes are on opposite sides. The nuclear membrane forms around each set of chromosomes, which in this case we've just got one set of chromosomes because their cell wall is so small. So nuclear membrane reformed. And here's a really important point about meiosis. During interkinesis, the DNA does not replicate. Because of this, the gametes that result from meiosis will only have a single copy of each chromosome. There's a second stage of meiosis called meiosis 2, but it's not much different from mitosis, which we've already seen. So I'll spare you the pain of going through it. But let's hear what meiosis really means to reproduction. All the members of the cellular cast will sing this song now.
Is that everyone in the cell wall or cell wall too?
It includes the cell wall as well. Meiosis.
See all the phases, and then
Keeps the thousand spheroids. It's much better than pure meiosis.
meiosis.
Now, whatever happened to those pigweeds? It appears that meiosis was successful and they were well on their way to producing several thousands of progeny. At this point, the cell wall can go away. The cell can go away. Now we need Palmer and Amaranth tell us what happened to those pigweeds.